สวัสดีครับ and welcome to Faculty of Tropical Medicine, Mahidol University. Today, I would like to welcome you, all participants and everyone, to our intensive one-week hand-on training course for bioinformaticians. This course is organized by Faculty of Tropical Medicine, Mahidol University, Thailand, and Wellcome Trust Sanger Institute from the United Kingdom. My name is Dilek Rimmathurasakul. I am Associate Professor at Faculty of Tropical Medicine, Mahidol University. I have been working in melioidosis, sepsis, and MI research for 20 years. We all know that the problem of the antimicrobial resistance, or AMR, is a big problem in the whole world, particularly among developing countries like our countries in Southeast Asia. This problem is caused by the bacteria that should be able to be killed with the antibiotics. However, overuse of the antibiotics causes the bacteria to gain the ability to survive out of it. It's a big problem because if you get the wounds or if you get to the hospital for the giving birth or surgery, more and more people might die of the infectious disease. Obviously, we all need to stop overuse of antibiotics. But the bacteria is already spread, already caused the infection, and we need to stop the outbreak of the bacteria that is resistant to the antibiotics. To be able to do that, the knowledge to understand the genes of each bacteria, whether the bacteria spread from someone to someone, or whether the hospital has an outbreak of the infection of the antimicrobial resistant bacteria or not. The knowledge of the understanding the genes of the bacteria is very important. Nowadays, we call the sequencing machine, or the machine that can understand the bacteria genes are cheaper and cheaper. The new one, even as small as a thumb drive, that can be used connected to one single computer. However, the big blockage of the bioinformatics is the analytic personnel. Bioinformaticians, which means the person who can do the computer analysis of the data to give the information or knowledge back to the director of the hospital, to the efficient control of the hospital, whether these genes is linked to those genes or not, is rare. They need to be well trained. Of course, there are a lot of master and PhD training courses in the world. But this opportunity to have a hand-on experience workshop from the world-class organization like a Wellcome Trust Sanger unit from the United Kingdom is a blessing. Therefore, we are so proud to be the host of this one-week intense hand-on meeting and workshop. Participants can come and learn how to do bioinformatics analysis from the real exercise, and they can be a trainer in the future. We hope that the knowledge that they gain can be used to analyze the real data in their hospitals or in their country, so that they can understand the mechanism of the antimicrobial resistance in their country and stop the outbreak of the infections. They can work together with the interventions of the National Action Plan on AMI in their own country. Overall, we hope to save more lives. Thank you very much.
Hi, so I'm Ewan Harrison. I'm a microbiologist based at the Wellcome Sanger Institute and the University of Cambridge. Sure, so the course that we're now running, this is the third time that we've run the course, the second time we've run it in Asia. The idea behind the course is obviously antimicrobial resistance is a major global problem, and there's a need for training all over the world in more scientists but particularly in the methods using genomes to understand how antimicrobial resistance is spreading, about transmission, about understanding about the evolution of resistance in general. So this course aims to train scientists from across Asia in methods for both the laboratory analysis of antimicrobial resistance, but also using genomes and bioinformatics. Well, you know, we're incredibly lucky to first of all to be able to run the course here, so we can really go into detail into the experimental methods, but also into the computational methods. And we've got a fantastic collection of young scientists from across Asia. And the real outcome is helping the next generation of scientists who are interested in researching, but also policy makers and doctors who are involved in antimicrobial resistance, helping them lead new research projects and develop new skills to tackle antimicrobial resistance.
Hi, I'm Dr. Liz Batty. I'm a bioinformatician and I work at the Mahidol Oxford Research Unit in Bangkok, Thailand. So the hope was that we could bring a course which had been developed and run in other countries and bring it into Thailand and be able to invite more participants from the region to come and join us and learn advanced skills in investigating antimicrobial resistance. So I did one of the first lectures for the course and it was a really great experience. The students are all very motivated, they're very engaged with each other, they're helping each other tackle some of the problems of the course, and it's been a real pleasure to teach them.
Hi, my name is Cassandra Sue. I'm from Welcome Connecting Science, where we aim to connect people with science. So on this course in Thailand, this is the AMR Asia course that we're currently running. And it's one of the initiatives that has been run for quite a few years right now. And it's part of our global training program. So what Welcome Connecting Science does is they basically fund global training programs in low middle income regions across the world so that we are able to facilitate the increase of genomics capacity in these regions. So for me, just personally, this is the first global training course that I've been on as part of the program, but so far I'm really pleased by the quality of the participants that we've got, and also the training team is excellent, and I really hope that the course participants will come out of the course having achieved all of their learning goals and also having learnt more about the field. And they'll be able to take it back to their relevant institutions and then move forward with their careers and also obviously teach others within their institutes what they've learned on the course.
Hello, my name is Claire Shiwapricha. I am Welcome International Intermediate Fellow, the Sangha International Fellow at Welcome Sangha Institute in the UK, and also a researcher based at Faculty of Tropical Medicine, Mahidol University, Thailand. Yes, of course. So AMR is a big global threat and is disproportionately happened with escalated AMR problems occur in Southeast Asia. The problem is we clearly need better genomic surveillance, but we currently lack the local capacity to perform a full genomic surveillance from basic microbiology to the genomic analysis. And this is why we need a Welcome Advanced course to help train and better the local capacity so we can cope with the problems better. I think all participants, they confront similar problems in their home countries. They witness the rise of AMR in the clinical settings, but the problem is we don't have tools to investigate this deeply. So what participants would learn from this course would be a new tools, a genetics analysis, and a way to integrate the data together to help answer the missing jigsaws in the AMR genomic surveillance.
My name is Pemjit Amonchai. I am a microbiologist, lab manager, and the area safety officer of the microbiology laboratory, Mahidol, Oxford University. I work with Mahidol, Oxford for a long time ago. I have the experience for about the laboratory, especially for the melioidosis, leptospirosis, and also the ESBL pathogen. I work with Molu for nearly 21 years, and our works in the field and also in the hospital, we have many colleagues that have collaborated with us in Thailand and also in around the world. It's very great and the good opportunity that I be here to be the lecturer and train the participant in this course. And this because I has been be the participant in the AMR course for a long time ago. But this year I have the chance to be the lecturer and the trainer to teach them about the AMR course. From my point of view, I think that the participant can get more experience, the skill and the technique that we teach them in the course. Especially they also can get the network and communication, because in the course not only in the Thai person, we also have the foreigner around the world that can join the course. So I think that in the future the AMR problem can be solved once we have the collaborate or the communication with friends in many countries. Okay, today I will bring you to the lab and see the atmosphere about the teaching and the course. Follow us. Yeah, this is the.
Hello, my name is Beth Blaine. I'm a researcher at the University of Cambridge. I've been there for about 10 years and I have a background in clinical microbiology. My main areas of focus are Staph aureus and Group B strep environment. Okay, it's been a real pleasure to teach the participants on this course. They've been very enthusiastic and keen to learn. They've taken part in all of the laboratory activities and they seem to have enjoyed it. I would hope that the participants they have a deeper understanding now of the importance of laboratory work for their future sequencing work, the importance of quality control and good lab work. And I would hope that they continue on with that and pass on that knowledge to their own colleagues.
Hello, I'm Janjila Thaipadung Phanit. I'm a member of Faculty Tropical Medicine, Mahidol University. I'm a microbiologist. I'm focusing on the infectious disease in tropical medicine. I'm focused on the infectious diagnosis and epidemiology. I use the whole genome sequencing analysis as a tool for epidemiology. I help them facilitate the place that we will prepare the lab, and then it's it's more like my grand experience to teach a lot of students in the lab, and then that it helps setting all the lab and the protocol. So I more like gain more experience how to teach them like interactively here, and yeah. I think participants will be fun and learn a lot of the AMR in both laboratory to test the drug resistance in the lab, and also use the bioinformatics to analyze the gene of the resistance in bacteria.
Hi everyone, so my name is Lalit Kumar. I am from India. So I am working in a hospital in India. So I am doing job as a junior research fellow in the hospital. So my background is like the biotechnology, but then I moved to in the microbiology. So in the first day, like we have just the introduction of the course, and just we learn about the AMR, what is the AMR and how we work for the AMR stewardship. And so we like I am just a beginner in the bioinformatics. So the trainer will give the best training. So they start with the basics command. So I have now hand experience in the commands. Also I have experience in the lab work. So I did the MIC and the AST with the Kirby disk method. And also I feel hand experience in the commands, do the assembly, do the fast QC. Also today I learned the other thing how we can do the LSM and convert to the BAM files. Also I am feel more experience in the field of the bioinformatics. I feel I will learn more during this course. So this course is beneficial for me, also for the other my lab members. So because I am working in a low middle income country, so there are no proper resources there for do the sequencing and no proper resources for the analysis part. So after getting this to learn these analysis and the AMR stewardship, so I will help or teach my lab mates or the lab members to how can you do the analysis, how can you do the the sequencing. Also you can do how you can make the AMR stewardship and what precaution you can take during when you are doing the MIC and the AST. Also it's help in the clinical perspective. Also we can control the community acquired or the hospital acquired infection during, because in our setting, so hospital infection is common. So but we also do to look on the community infection, because the AMR has the role in both the community and also the hospital infection. So with the help of to gain the knowledge in the AMR, so we can look into what's the role of ARG, how can AMR transfer between the humans, animals. So it's important for both the clinical view and also for the community view. Thank you.
Hi, I am Nika, a researcher from the University of the Philippines. I am very thankful for this golden opportunity given to me to be a part of the AMR in Bacterial Pathogens, Thailand. So first off, truthfully, I am a beginner in bioinformatics, but this course so far has really helped me a lot in appreciating, understanding, and even improving my skills on bioinformatics. All of our teachers, from the lecturers, laboratory, and even the computer instructors, are very helpful. They also have high knowledge and are very effective in teaching every one of us to improve our understanding of bioinformatics. So I hope I can really apply this when I come back to my country and do our research on AMR surveillance. This course has been very fun, and I learned a lot, not only from our mentors but even my co-participants. So we come from different backgrounds, different countries, and different areas of expertise. So every day is really a new learning experience for every one of us. This course is also very high put, very high yield. We learn a lot from the lectures and the hands-on exercises. The highlights of the course for me is learning about diagnostic surveillance. And before, I was afraid of doing bioinformatics, but now I can say that the virtual box and the command lines are my friends. And I really hope that in the future, when I do my research on AMR surveillance, I can use these tools that I learned from this course.
I'm Ratanin. I'm a PhD student from the Department of Microbiology and Immunology, Faculty of Tropical Medicine, Mahidol University. I'm interested in bioinformatic analysis because I work on whole genome sequencing data of Bacterial Pathogens, a bacterial pathogen that causes melioidosis. I use whole genome sequencing data to study the epidemiology and pathogenesis of this pathogen. And actually I'm a biologist. I have a little bit experience about bioinformatics, and I found that this course helped me a lot to improve my knowledge and my experience, and then I can use it to complete my thesis project. So I finished some part, but I still need some more skill to finish it. And I also found that when working by doing, like me, working by doing, sometimes I learn by myself to finish my work, and my knowledge and my experience is a little bit jump. It's not start from the beginning, start to step 1, step 2. So from this course I can get step by step for bioinformatic analysis, so it's helpful. Yes, I enjoy it, especially bioinformatic tool to detect antibiotic resistant genes. I never experienced it before, so my work previously focused on the phylogenetic tree analysis, or to study the population of bacterial pathogen, but this is my first time to use bioinformatic tool to detect antimicrobial resistant genes. Okay, yes, my bacteria that I have worked now is not resistant. I mean, it's not one of the most important bacteria that resistant to antibiotic, but they also resistant to some beta-lactamase, something like that, and some recent study, they found the mutation that caused the resistance in this bacteria. So I think this will be useful for me to work with this bacteria, and also can be applied with other bacteria in the future.